Yes, ma'am.
Okay. Okay, shall we start? Have you all finished your previous experiments?
Yes, ma'am.
Okay, does anybody facing any kind of problem in like when performing any experiments?
Ma'am,
Yes, Sarna.
ma'am, in the T Blastex, I got like MBPT results, but like they were mentioned, it was mentioned that there are no matches, something like that. Ma'am, can I just show the screenshot once?
Yes, yes, yes.
Ma'am, this one I got
Okay,
for.
wait, hold me first, please. For everybody, are you getting the same results or like different kind of results? Anybody can tell? T blast
Ma'am,
X,
this is
right?
the result we get actually.
This is the result?
Yes, ma'am.
This is the result we get. There is no similarity,
Okay, no
Raj.
problem. That is also fine. Like if you're not getting any hits, also that is fine. If still, if you don't want that ID, then I can change the ID to you people. Is it okay?
Okay,
Ma'am,
ma'am.
I think
I
it is okay. Yes, ma'am.
Yeah, because now that is also a result actually. Like for that protein, you will not having any similar kind of things. That's why it is showing like that. There is no hit found in that one. Okay, that is the result. Like it's not like you're not getting results. You're getting results, but you have to interpret it like we are getting like for this protein, we are not having any similar kind of thing in blast. Like and the database you have chosen which one like that because I didn't perform that for that one. I was just giving you the IDs. I didn't perform for that one. Okay, let me check that one. Any one of you can send your like finished part to me. Then later I will check it and tell you. Okay, if any changes it might need, then I will tell you again. Okay.
Okay, ma'am.
Okay. Up to we have seen what else we have. Okay, we have finished up to 4 experiments, right? Uh,
Yes, ma'am.
Last we have seen up to using faster tool as well as like local alignment and global alignment in emboss. Then what else is we can cover today? And dot matcher. We'll cover these three things, like these four things today. Is it fine?
Yes, ma'am.
Yes, ma'am.
Okay. Okay, first let's proceed with the FASTA tool. Actually, a tool is there which is like FASTA we used to study, right? Generally, we used to read about FASTA. This kind of thing is happening in this FASTA formats are there, and for that, a special tool is there in Emboss package. I will share that screen with you. We'll just see. Okay, this is a like blast we will do, right? The similar type of thing we can do in like fast also to find out the similarity between our searchers. Here they have given you options. So if you see here, this tool provides you a sequence similarity searching against foreign database using FASTA suite programs. And it is having a global query search and local databases, and these things they are having as an optimum temperatures. So if you see the databases, what are the things they are having in the sense? Uniprot KP, like that is like manually curated here, and mostly they are having because this is for 14 searches, na? So we are having for different things also. For nucleotide also we are having, and for that we are having DNA coding sequences. It has been like um, European Nucleotide Archive. That is ENA belongs to that EMBL, and it's taking sequences from that place. And genomes also we are having, and for proteomes also we are having. So for your work, we'll go with the protein similarity search, and we will search against UniProt. If you are finding any similar kind of protein in that one or not, let's take it to like this FASTA file. We need to paste our FASTA sequence. So I'm pasting a certain protease from this one. Like okay, let me take the sequence. Wait a minute. Yeah, I have taken the serine protease from Anopheles. Let's check it for the similarity search sequences to FASTA. Like it's a similar kind of thing. Like whatever you have done in BLAST, right? You will be getting almost a proper similar type of result, but using a different tool. You just need to know how the different tools are working. That is the main motto of this thing. Like already you have done this in BLAST. The same thing you are doing again to check out like how this tool is working, what kind of things they are giving. So we have given that sequence input as like from Anopheles. If you see here, they have given you like similar proteins of like from different species with the length and score bits and identities of like how much and positivities and its e-value. Like lower the e-value, the best will be the like identity. You see here e-values are around 5.1 and 4.0, and the identity is also a little bit less, like 32. If it is like exactly 99% of matching in the sense, then the e-value will be the zero level. Uh, you see they have given you up to 50 results. For this experiment, you don't need to do any like much studies and all in this one. I'll just give you a protein ID and like how to you just title like experiment number you name it as 5, and put day to day and make it as a faster tool and perform pairwise element of protein sequences, like protein sequence, whatever I am giving with accession ID using faster. Then tool used, in that tool used you give faster, and with the source code you will use this like URL and procedure like ABI page, like going to the emboss page and searching against the faster tool and paste the protein query sequence and submit it. That is the main thing you are going to do in this faster tool. That's not much explanation needed for this one because this is almost similar, like no, exactly similar like our blast tool. We are finding like homologous sequences between our proteins, right? And accession ID I will give you. You just search for that one, and okay, I will give you the accession ID. Can you note it down? Okay, let me type in the comment box. This is accession ID for this faster tool, and this Homo sapiens seren protease. You can just check for that one and then just paste the sequence. That is enough in this experiment. Okay, is it clear? It's a very simple experiment because you have already done this thing several times. That's why I'm not explaining you about that. Is it fine, or do I need to explain more?
Yes, ma'am. Ma'am, we can proceed.
Okay. You just make a note of that ID alone. Then place your outputs out there, and then just like interpret your results. Like how much identity it is there with this one, like how many sequences we have got, like that kind of information is enough in this one. And okay, let's go to the pairwise alignment. Like this is also a pairwise alignment search tool, and like now we will go for like identifying the relation between the two sequences using a dot plot. You know, you guys have already studied about dot plot. Have you
Yes,
all
ma'am.
have any idea? Yeah, that is also a similar kind of thing what we are doing here. And generally, if we are let me show some PDF once again. Can you able to see visualize this page? Okay.
Yes, ma'am.
Yes. If you see here, like generally we used to say dot plot as like if we are analyzing two sequences or two sets of ABCD or something, in matrix we will match, right? Like if it is matching, we will put a dot. If it is not matching, we will leave it as it is. So likewise, if you see, this is an example for that one. Like I'm having a sequence called DIL in the x-axis and IVQ in the y-axis. I'm trying to match manually with this thing. Like I and I are matching, so I'm putting a dot, and D and D are matching, I'm putting a dot. Like this, I am matching. This is manually I'm doing if I have a very less number of sequences. So what will happen if I have sequence of around like protein sequence if I'm having 1,000 and another protein I'm having it's for some 500. So is it possible for me to do it manually? So that will be a bit difficult. So we are checking like it's a relation between identifying the two sequences, this kind of domains. Those things we can check through this dot match. If you see here, it can the diagonal can be formed in this one, like a tilted one, two, three. This D, E, Q. The last three things can make a diagonal, but the other things cannot make a diagonal. If this is making a diagonal, which is exactly matching with these things, the EQ at the end and the EQ at the y-axis also, it is exactly matching. That's why we can make a diagonal out of here, and we can say this is like this region is exactly matching with this one, like A sequence and B sequence, like these two regions are exactly matching. It might be a domain part or it might be a motif region, like that we can control it to them. So now for five, just here are some six residues are there. So what will be the thing, like if we have more than 1,000 residues, or in that case how we will do, like we are having already we are having for those things we are already having all the algorithms that have been Like there, now we'll see a tool called dot matcher. There we'll try to paste two sequences, some different species. You're going to give me the ID. I just think about some protein ID or something, and we'll see how it is working. Okay. Be ready with your any kind of protein names. Whoever is willing to share, you can share your like names.
RV0183.
Wait, giving your ID, right?
Yes, ma'am. Protein name, ma'am.
Protein name. Okay.
Yeah.
Say your protein name.
RV0183.
RV0.
183.
Okay, this is monocyte glycerol
Yes,
lipase.
ma'am.
It's type.
Mycobacterium.
Okay, from Mycobacterium tuberculosis. Possible phospholipids. Okay, I am taking a sequence of this one. Some other sequence from different. Okay, we'll try to get. Somebody can say any other like protein.
Some protease.
Some protease. Okay, let's try go for that one. I'm pasting for sequence here. Uh, this is from possible live phospholipase from Mycobacterium tuberculosis H3R37RV gene. Okay, and then another sequence I will try to paste. Let's wait a few minutes. Okay, we'll try to paste it to different organism. The same, or we'll go for some other protease. Okay, I have pasted that sequence also. Like that is a protease from human rhinovirus, and this is a lipase from Mycobacterium. This is a dot matcher, right? So it will match each and every amino acid, whether it is like having a matching pattern or not. Okay, we'll try to submit this one. And threshold level and all like all are same for those things. Want to do any changes in the threshold level? Okay, if you see, okay, we got the results here. There is no exact matches between these two sequences. It's because this is a lipase and this is a protease, right? One is lipase and one is protease. Only this region, like around, and this is from 400 amino acids and this is from 290 or some amino acids in that one. Mm, okay, this region is alone matching. This region, it's been like totally like evolutionary. It has been two from each other. This lipase and protease, because this is from mycobacterium, this is from like human rhinovirus, right? These two are totally like opposite one, and we are having only a short context. These regions we used to say it as a context because only a bit matching is there. Like here it will be there, but in some other graphs you can see. Like I will make one more search also in that one. We'll see whether we are getting in that one or not. These regions are called as context. This context, what it will have in the sense, it will be having a specific type of domain or specific type of motif, which may be applicable to all other lipases or all other proteases. These kind of like motif regions are there, right? It can be a like functional region which is present in that sequence. Now I will try with a different sequence. It's this two or not like making any like matches. So I will be making the same. Okay, I will paste another set of sequence with one. Like let it be like the lipase. Let it be like that, and I will change the other sequence alone. this is an hypothetical protein from Z maze. Somebody's mic is on. Okay. Okay. If you see here, we are getting some set of. Is it visible? Yes. This is from Z maze, and another this is from Z maze. It's also lipase protein, and this is from mycobacterium. That is also a lipase protein. We see these both are lipase protein, and it is having a similar kind of matching in this region. So that's why it is making a diagonal. See, but the diagonals are breaking in between, which means these regions are not matching. Only the regions which is matching. Now, if it is making an exact diagonal, then we'll say these two proteins are exactly matching with each other, and it is having its highly conserved residues are there in this one, like that we can say. But here it is not highly conserved, and if you see here, this is a short contact region which we have found in this one, and this short contact also it might be a functional part, like similar part um, for these two proteins. Okay, like if I am matching the same sequence with same thing, we can find out the exact matching. See, we are able to see this is like I have pasted the two sequences, the same sequences, and it is exactly matching with the short contexts. This is just to show you like how it will form if it is exactly matching. Just to show this thing. For you, you should not make it like this. You should make it like the previous one, like with two different sequences. You have to make your dot matcher, and you have to show your results in like that. Okay. Okay. I will give you the. Sorry. For these two things, you have to make with the different sequences. I will give you an ID for those two things. It's from insulin. Okay, I will give you like down the ID in comment box. You just note it down for this experiment. This is an experiment number 6. Pairwise alignment. Heading is pairwise alignment. And in the subtopic, you have to make it as the first one as a dot plot. In the dot plot, you have to perform for these two things. I will give you the ID. Can anybody perform now and show the results? Is it possible?
Then what you said about subheading?
For writing.
Yes, ma'am.
Yeah, this is like sixth experiment. Pairwise alignment. It's like a topic as pairwise alignment. Okay, and as a subheading, you just put this as a dot plot. Okay, and in dot plot, you have to show the results of these two things, like whatever I have given, like giving you now. You just make these results. Okay. If somebody can finish this work and show it to me, the results. Like, can you share the page? These two are insulin proteins. One is from Octodon Dagus and another is from Danio Radio. Whoever is performing, you can show, like after finish, you can show it to me.
And connect with
Okay, sure.
And is it visible?
Yes, yes. Okay.
Here it is. Yeah.
Okay, yeah, this is like, yeah, this is the answer. Now, if you see here, we are having two different, we are having a diagonal line which has been separated in between. One is from, like, both are from insulin, but it is having exact matches in these regions, but not on the other regions. So, like, can you able to understand this graph?
Yes, ma'am.
Is it clear
Yeah.
Yes, ma'am.
for everybody? Yes. Okay, this is both of them. One is from Octodon Vegas and another is from Daniel Rero.
Yes, ma'am.
Okay, you just save this result for your record work. Okay,
Yeah.
no need to do again. You just save this result. Okay, now like this is the first topic is the dot match, right? Then next subtopic is the global alignment. Okay, global alignment.
Ma'am, shall I stop the series?
Yes, yes.
And
Thank you. Okay, first subtopic is like dot plot, right? Then we'll be seeing about in global alignment and a local alignment. We'll be seeing in the further sessions. Okay. Wait a minute. What? Okay, shall we proceed with global alignment? So, okay, what is the
Yes,
difference between global and local alignment? Can anybody explain? Like, I think you have already studied in your paper, like theory paper. Can anybody tell like what is the difference between these two things? Like, no need to like elaborate it, just to know whether you guys have an idea or not. No need to worry. Like, if you don't know, also it's not a big deal.
yes, ma'am. Actually, these are like alignment kind of
Hmm.
thing. I mean, for performing similar to find similarities between two sequences, and global alignment it tries to like the algorithm tries to match from end to end of the both sequences
Okay.
globally. So that term global
Okay.
comes from there only. Then local is like highly conserved region or particular
Yes. Wherever it
from
is
I
like
mean from
starts
one sequence.
matching from
Yes,
that
ma'am.
part, it will like
It
start
is
match
not actually
aligning.
from end to end. It is
Yes.
particular to the particular region.
Okay, that's so fine. Okay, let's see, like have a thing like how we need to perform this both local and global alignment using MOS. Like we are having two algorithms called Engelman Wunsch algorithm and Smith-Waterman algorithm. So Smith-Waterman algorithm we will be using for local alignment and Engelman Wunsch algorithm will be using for global alignment. Okay, so these two packets are already existing in the MOS. If you see parallel sequence and having a two set of like alignments for global alignment, we are using Needleman Wunsch, and for local alignment, we are using Smith-Waterman algorithm. These two algorithms can does anybody like having any doubt in this one? You can ask me in your free time. Like I will try to teach the like algorithm part. For right now, I will be teaching only the tool how to access this tool. Okay. In your free time, you can ask me if you're having still having doubt in like how it's working in at the back. Okay. Okay, we'll go for this Needleman Wunsch algorithm. This alignment, okay, we'll use same set of sequences for global and local alignment and see what is the difference between these two sequences. Like I'll be pasting a set of sequences for global alignment. Same wise, I will be pasting those two sequences for local alignment and we'll see the difference between these two alignments. Okay, that's what we are going to do now. Wait a minute, I will be pasting the sequences in a minute.
Then which tool you are using, ma'am?
This is from Emboss. This is for global element Needleman Wunsch algorithm. Is it visible? Like Emboss Needleman Wunsch algorithm. It's from Emboss. Is it clear? Okay, I will be pasting the sequences. Like, wait a minute, I will search for the sequence.
Ma'am, same sequence you are going to use or different?
Yeah, I'm going the same set of sequences to show you the variations, like how it is like aligning in global element and how it both okay, let me say it clear and again, like wait a minute. Here, this is a parallel alignment, right? We are using two algorithms, Needleman-Wunsch algorithm and another algorithm is Smith-Waterman algorithm. Smith-Waterman algorithm is for local alignment and Needleman-Wunsch is for like global alignment, right? So how the Needleman-Wunsch algorithm works, it will be start matching from like end to end, in the sense from the beginning it will start aligning till the end. But for local element, it is not like that. In local element, it will start to match the exact matches. If it is in between, also it will try to do from that place. Even if it is at the end, also it will try to do. It will leave the first part, like whichever. If for example, if I'm matching 500, first sequence is having 500 amino acids, and my second sequence is having some 300 amino acids. So it starts aligning. I'm using this local alignment, and whenever it starts aligning now, so what it will do, the beginning region, like if it is not matching with the first 100 amino acids, so it will leave that part. I will start aligning from 101. So that's how the local element will work. But in case of that global element, it is not like that. It will try to start align from the beginning. Even if it is like mismatches or like gaps, also it will increase the gaps and the values will be different. If you check the identical percentage between these two things, local element will be having a high percentage of identity than a global element, because in global element more number of gaps will be there, more number of extensions will be there. To try to make it like a proper alignment, it will include all those things. That's why the global element will be having a very lesser identity sequence than a local alignment. Like, is it clear? Okay, while showing the results you will get to know like how it is working. Okay. Okay, so the one is asked from which species? Okay, let me see that one. I forgot the name. And one is from Untitled Chapter. Okay, we have got the results. Okay, you see, this is a like this algorithm like Binf algorithm. Here we are aligning, trying to align two sequences of this one. This NP and this XP. They have given you like opening penalty as 10 and extension penalty is 0.5, and in the penalty they have given the matrices it used E plus and 62. Those cavities are there in the first part, and they align the sequences. Here you see we are having the length and identity and similarity. So length is 924, and identity is like 42%, 42 like 4.5 percentage of sequences are like identical, and similarity is 8.7 percentage, and gaps are around 80.7 percentage. If you see here, it will start aligning from the first region because it's a global alignment. Here there is no matching, so what it will do, it will give you the gap opening and gap extensions. The gap extension is continuing up to this much. After reaching this region only, around after 251 residues, it's try to align with the first sequence, like first amino acid with the second sequence. It's try to make the maximum alignment with these things. So if you see here, MM and MR matching, it is exactly matching in the sense it will give you a line. And if T and C are there, like semi-conserved, I think I've already discussed you about these things. If two dots are there, what it will means, and single dots are there, what it means. If two dots are there, it might be from similar group of amino acids. If it is a single dot, then it will be a semi-conserved amino acids. Okay, the functional group will be same. If two dots are there, then in that region, the function of those two amino acids will be almost similar. If both are acidic in nature, then that will be mentioning in like two dots, like glutamic acid and what else is there? G and N, E and N, like the two amino acids. And this for aspartic acid. Yeah, D is for aspartic acid. The aspartic acid and glutamic acid are both are having acidic property. If those two are matching in the sense if it's coming in a matching line, then it will be mentioned as a two dots because its property is same. That's why here some two dots are there and some single dots are there. Single dots are called as semi-conserved regions. So only at this region only it is from 251 to 475 region of this region only matching. Then the other rest of the regions are all totally unmatching. That's why its similarity is around 4.5%. The same for the same sequences, I have done water algorithm with Smith water algorithms. If you see here, is it visible?
No, ma'am.
No, ma'am.
Could you please share the ID
Okay.
so that we could try it now?
Oh, for Needleman virtual cell is not visible.
Now, ma'am, it is visible.
This is water algorithm. Okay.
Yeah.
You see here, this is embossed water. The same sequence I have applied those two same sequences. Then I'm getting in a percentage of identity of 21 percentage, 0.7 percentage. There I got around what I got around 4.5 percentage, and the similarity of around 8.4
8%.
percentage. Yeah, and the gaps around 80.7 percentage. But if you see in that Waterman like Smith Waterman algorithm, like everything has been like the gaps has been reduced because we are not considering the like unmatching parts. If you're not considering the unmatching parts, then the gaps, like the values for those gaps regions are there, right? Those will be like neglected. At the end region also, those regions will be neglected. At the beginning region also, those like whichever is not matching, it has been neglected. So only the rest of the things has been like matched with each other, and the score it is around 21.7 percentage. So like when we will be doing this, I will give you a question like at what time we will be trying to do global alignment, at what point we'll be trying to do local alignment. Do you have any idea? You have to find answer for this thing. I have teach you this like how to perform these two things. Then in which situation you will go for global and which situation you will go for local.
Ma'am, if you want
Whatever
to
is
find any conserved regions, we will go for
yes,
local alignment between two
yes.
sequences.
Then for global, if you just want to know like how much it is evolutionarily like distant from each other, we'll go for Global. Okay.
global. If we don't know about the
Mm.
information.
Yes, then these are the two different cases we will choose. Like whenever you're choosing an alignment tool, you have to be very clear about that thing, like which tool you're going to use and why you're going to use that thing. If you're choosing local alignment, then you should tell them like I want to identify some conserved regions which is functionally active in my protein. So that's why I'm going for local alignment. Okay, between these two sequences. If you are having, I want to check, for example, imagine you want to check your, okay, you are checking your hemoglobin protein, which is highly conserved with mouse or something. We'll go for mouse. So for both, we'll be checking a local alignment because the sequence number may be vary. For maybe for mouse, it will be having some 500 sequence, 500 base pairs. Like sorry, base pairs, it's an amino acids, 500 amino acids. And for human, just imagine we are having some 1000 amino acids for hemoglobin proteins. But both are having similar property. We know that it is having a similar property, but we don't know which region is exactly aligning and which region is making the interaction between the ion. Iron, right? So what we will do, we will try to make an alignment for those regions, and we may find a hit in between 300 to 500 amino acids, 300 to 400 or 300 to 350 amino acids. Those region is like an exact matching region where your heme group can go and bind to your hemoglobin protein. Okay, can you able to imagine the thing? So that's why we are going for that local alignment. So if in that case, for example, this region is like your heme binding region. This region is exactly matching in the sense, like this region is exactly matching in the sense. This is a highly conserved region between these two sequences, and the same kind of pattern is there in mouse. So we can proceed our work in mouse because in human we cannot do all our wet lab things with the human, right? So we can go for this mouse and we can try to imitate the region like as it is in human, and we can perform our like what kind of work we want to do. Those kind of things we can do with the help of local alignment. Is it fine? Like how we need to do this global and local alignment?
Yes, ma'am.
Yes. And in for this tab is for like global alignment. Like the identity is very less. See, it's 4.5. So it's not optimal for so many conditions. It might be totally like distant amino acids from each other. And the identity and similarity, even if the identity is not there, that similarity and the gap score will be the gap score is high because we are neglecting the gap scores there in that local element, but here we are applying that gap scores like extension and opening penalty we are giving. That's why the value for gaps are a bit higher in global element. Is it okay? Shall I give you the IDs to perform these two things?
Yes, ma'am.
And the Network is slow a bit, just wait for a minute. Okay, next I have sent you right, like those two proteins. One is from hemoglobin of which species, and another is from Clostridium, that is a sortes. We'll try to identify any kind of similarities there or not in between these two sequences. Whoever like finish this thing, you can share it with me. Okay, I will give you 10 minutes of time to try to prepare those things and then just tell me.
Ma'am,
Yes.
ma'am. So for both algorithm, like for both elements, we have to use this ID, right?
Yeah,
The
both are
record
because
also.
in record also that
Okay.
then only you'll be able to see the difference between these two things. Like in global, it is making this much of percentage of like matching, and in local, it is making this much of percentage of error. That's why I'm telling you to see the difference between these two things. Okay?
Sure, ma'am.
Yes. You try and let's show it to me.
Ma'am, we have to do both, no?
Yes.
Okay, ma'am, completed. Can you?
Okay. Can you show it to me?
Yes, ma'am. Is it visual, ma'am?
Yeah. Can you zoom in a bit? Like,
Ma'am, I'm using Linux, so
okay, no problem. Okay, this is having the 22% page of
yeah,
like
I didn't.
local element, right? Can you show the next tab of
Yeah,
global?
yes, ma'am.
Okay, how much it is showing?
0.5, ma'am.
0.5. So nothing is matching in that one, right?
Yeah.
Okay, only a short set of regions has been matching
Mm-hmm.
in that one. So can you able to see the difference between that local and global?
Yes, ma'am.
Okay, you can save these results for your
Ma'am, can we add multiple sequence in the global alignment? Yes.
no global. This is a pairwise alignment, right? In
Okay.
pairwise alignment, like we can do for two sequences. In multiple sequence element, it will be having a different algorithm where it will match like AB. If you are having ABC,
Mm-hmm.
first it will match with AB and then it will match with C. Like that, it will go. in multiple sequence alignment. In pairwise, we can only go for 2 alignments.
Okay, so ma'am, in the alignment case,
Yes.
here is a match only, right? This
Yes,
one.
yes. Hmm.
Okay.
Okay, this region we can say it as a short motif region.
Yeah.
So only some 5 residues have been mentioned right in
Mhm.
that region. Like some what is it 80? I couldn't able to see the thing. Like it's not visible for me. Maybe some 5 amino acids is matching, right?
Ma'am, is it
Uh,
visible now?
yeah, some VL. Okay,
SC?
VT. Yes. So we can actually it is not exactly it might be a motif region or not. Okay, if it is a motif region, for example, in some bacteria, like we will be having a specific motif region called LPXTG, we'll say, which means like at the end region, it will be carrying out those 5 amino acids together in all the proteins. The like whatever I'm telling, like that LPXTG motif that belongs to a cell wall anchoring protein. So wherever cell wall anchoring protein in the sense, it will hang into the cell wall of the bacteria, right?
Okay.
Mm. So it will be hanging there. Whatever bacteria it is having that cell wall anchoring proteins, those will be having that LPXCG motif region. So
Okay.
we can make a like identification in that one. Like if it is having an LPXCG motif, so we can say okay, this is a cell wall anchoring protein. Like it's a short set of sequence. If it is around 20, more than 10 amino acids or like 15 amino acids, then we'll go for that as a domain. So you should know the difference between that motif and a domain. Motif is a short set of like short set of amino acids around 5 between like 10 in between 10. It should be there not more than 10. That will be a motif region, and if it is more than that, then it will be say it as a domain region.
Ma'am,
Okay.
here, if we see carefully here,
Yes.
so there is a two dots,
Yeah.
okay, and one dot. So what does it mean, ma'am?
Like two dots in the sense like V and I are matching, right? Isoleucine and valine are matching. In the sense, it might belongs to a same set of amino acid group. Like
Okay.
what kind polar amino acid groups, valine and isoleucine are present in the polar amino acids, right?
Okay.
So we will be having a similar property. Like all the polar amino acids are having similar properties. But if it is single dot, it is there in the sense is with like serine with isoleucine, right?
Mhm.
Yeah, serine with isoleucine is a semi-conserved semi-conserved amino acid, where if we are removing any one kind of one group, if I am removing one CH3 group, it can make it to a isoleucine. Or if I am adding one kind of one group of C, then it will make it to a like semi-conserved region. That will be mentioned as like single dots, and the double dots will mention the like similar group of amino acids.
Okay. Okay, ma'am. Got it, ma'am.
Yes.
Mm.
Then who else has finished except Babu? Like
I have finished.
yeah, okay. Subhash has finished. And anybody else
Yes,
other than
ma'am.
Subhash?
Completed, ma'am. Completed.
Completed. Can any girl show your screen? Like Meha, can you share your screen? Is it possible for you? Or you're listening through mobile or something?
Ma'am, this experiment I'm doing on mobile.
Is it possible for you to do it in mobile?
I'm opening another tab and trying, ma'am.
Okay, okay, okay. Then try Chandini Devi finished. Chandini, she's not in class or okay. Let's remind Chandini later. You guys just remind her to do the experiment. Okay. Then Nishi, have you finished?
Ma'am, my system is not working now, so
Okay, just listening.
yes, ma'am.
Okay, try to finish
I
it
will do
soon.
it later, ma'am.
Yeah, try to finish it soon. If you are facing any problem, you just remind me, okay?
Okay, ma'am. Thank you.
Okay, you just remind Chandini to be in class for the next time.
Okay, ma'am.
It's clear, both these two experiments?
Yes, ma'am.
Okay. And while writing your results, you have to like mention like in interpreting results, you have to give your like identity values and the organisms we have choose. Like in the ID itself, you can like there will be there, right? From like what kind of protein it is and from which organism we have chosen the thing. Those two you have to mention, and whether it is homologous or it is distantly related, you can write your own in your own words. And like gaps, discuss about the gaps also. We are having this much of gaps in this global element and not in this local element. Like this, you have to interpret in your results. Okay, this is up to experiment number 6. Mm, okay, still we are having like multiple sequence alignment and second set prediction methods, binding site identification, sequence. Okay, we are having around 13 experiments, 13 or 12 experiments, I guess. Okay, let me check your syllabus once. Wait a minute. Okay. Sorry. I think here it is. When is your exam? Like at the semester exam? 24th December, right?
Yes, ma'am. December.
Okay. Okay. December end or in the beginning?
Ma'am, maybe from death.
From 20th. Like that's theory, right? Like or like practicals also will happen during that time, or it will before. It will happen before.
No, ma'am. First, then lab, then project.
Okay. First theory, then lab, then project. Because to check out like whether I will be available or not, I need to finish your syllabus. No, that's why I'm asking you. And I see. So not too many things are there. We are just having in multiple element. Now we will be seeing class W, uh, cluster W using cluster W, dialign, and multi. The three things, and in primary and secondary structure prediction methods also we will be using the GeoR, Cycled, and Chopasson method. Okay, can we see these things in next class? Like experiment number like 4, not experiment number, like what is number 6 and 8. Can we see it in next class?
Okay, ma'am.
Because not many things are there. Only like this multiple element is there, and second section population is there, and sequence profiles and patterns, protein motifs. Those things you need to do. Like ProSite and can even go in to see the databases. Is it fine for you, or should I take alignment, sequence alignment? Whichever way you want, I'll choose according to your wish.
Ma'am,
Ma'am,
next
next
class.
class.
Okay, then happy Diwali.
Ma'am.